This is the third video in the series where we'll find Adam, Eve, and Noah. In part one, we found that evolution is not true. We applied the scientific method to evolution theory and found its predictions false, and so evolution is not true, and in fact it's not even yet a science. In part two, we looked at the first chapter of Genesis for the history of biblical creation, and we found that there's scientific support for it. 6 days of creation are literally that when measured on the background radiation cosmic clock, and fossil record is fully explained when we look at biblical creation in that way. What we'll find today is Adam, Eve, and Noah. In chapter 1, verses 26 and 27 of Genesis, it says that God made Adam and Eve. And in the next verse, God told them to be fruitful and multiply and have dominion over the earth. The question we're going to ask in this part is there scientific support for the existence of Adam and Eve? We have not found their graves. We have not found the Garden of Eden. So we don't have any physical proof along those lines, but there is proof available, at least in terms of the timing when Adam and Eve existed on Earth. And to answer this question, we're going to have to look at DNA. DNA, which is deoxyribonucleic acid, is something you probably have all heard of. It is a basically building block of the information about who you are within each one of your 100 trillion cells. And we'll look at how it consists of chromosomes and genes and how the net result of all that is to give us a way to find when Adam, Eve, and Noah existed. Now, the DNA within your body exists in two parts. The first and largest part exists within the nucleus of each one of your cells. Now, we've seen this before in part one, where we have a cell outline here, where we have a zone between the cell wall and the nucleus, and then we have all kinds of activities that we saw in part one that were going on inside of the nucleus. One of the materials that's also within that nucleus is the DNA that determines your individual characteristics. I've shown here a very small part of one chromosome of DNA. It is a lot more complicated than I've pictured here, and there are actually, as we'll see in a minute, 46 of these within each one of your cells that define your nuclear DNA. There is an additional source of DNA which is outside the nucleus but inside the cell wall. We call that mitochondrial DNA. Now, those two forms of DNA, the nuclear DNA is the largest. It's divided into chromosomes, we'll see in a minute, and there's one particular chromosome in there called the Y chromosome that is donated father to son, and its important characteristics are that it rarely changes. We'll see that most of the other DNA changes slightly, but the Y chromosome changes rarely. Very similarly, the mitochondrial DNA, which is given from mother to all children, is small, and it also rarely changes. It's about the same rate that Y chromosome changes. We'll look at those two aspects of DNA, and they will ultimately give us a genetic clock to see when Adam and Eve, and as it turns out, Noah lived on Earth. Let's look at the structure of the DNA. First of all, DNA is a double helix. It consists of these things here that we'll call nucleotides, or the geneticists call nucleotides. These parts are strung together in two necklaces and two intertwined structures here, with one being the opposite in a sense of the other that I'll show you in a minute. But there's a so-called helical structure with one string wrapped around another. Now, a couple of things are important about these building blocks, and from now on let me just call them beads. Each one of these beads exists and can exist in only 4 different colors. So here we have an auburn one, a tan one, a green one, and a yellow one. So those are the only 4 types of beads that exist in your DNA, and each one of those colors corresponds to a particular chemical compound. The first letter of the color I've set here to be the consistent with, or give me a clue to the name of what that is. This yellow bead is actually canary yellow, starting with a C, and it is representative of the compound cytosine. Now these compounds come in two different versions, the same color but two different versions, and some are left-handed and some are right-handed. In your DNA, the only type that is used to form these strings of beads are the left-handed beads. So we've got all left-handed beads here, and it turns out that they all need to be either facing up or down. Let's just say they're facing up, such that for this auburn bead here, there's the up direction, there's the bottom part of it. The bottom part of that auburn bead is connected to the top of this tan bead here. And so they're all hooked up in that way, top to bottom, bottom to top, and they're all left-handed. And the further and final thing is that whenever you see a yellow on one string, you'll see a green seemingly connected with it. Or whenever you see a tan on one string, you'll see an auburn connected with it. And that happens with both strings. So those are the rules in which this DNA is put together. And the DNA, being this double helix string with 4 types of building blocks and 2 different things, each one of those strings contains the equivalent information. They're in a sense invert or inverse of each other, but they do contain the same information if there's the information that you need. And the information is a blueprint to make every part of your body. Every part of your body can be made from the DNA contained in every single cell in your body. Now, our human DNA within an individual cell consists of about 3 billion building blocks, or 3 billion beads. And small differences in the orientation or the order of those beads within your body determine each of your individual characteristics, and determine that each one of us have different, slightly different DNA. That's why they can determine one person versus another in terms of getting a DNA sample. Now, with 100 trillion cells in your body and 3 billion cells in each one of beads in each one of those cells, you have 300 sextillion beads in your body. That's a large quantity. Sextillion is 10 to the 21st power, an unimaginably large number. But to put that in perspective, you have about twice as many beads in your body as there are stars in the entire universe. There are about 150 sextillion stars in the universe, an unbelievably large number, but you have yet a larger number of beads in each one of your individual bodies. So that's just a very large number. Now, you might expect, if you were believed in evolution, or if you just thought about one of the consequences of evolution, it ought to be that if we are the most advanced living creatures in the evolutionary tree, it would seem likely, with all of our extra complexity, that we'd have the most complicated or the largest DNA component. As I said, we have 3 billion building blocks in ours, but yet we are not the largest. The largest genome, and the genome is the name given to the sum total of the building blocks and beads that you have in a given species DNA. The largest found today is a little water bug called the Daphnia pulex. It's about 1 millimeter long, and you may have seen still water and seen these little bugs, these little water bugs flitting along the surface of the water. They quite likely could have been the Daphnia pulex. And it has 4 billion building blocks. So here is a simple water bug that has far more, third more DNA information than we humans. Let's look at the duplication or reproduction process that goes on with DNA in the cells. As an original cell is ready to die, it knows it, and the first thing it does, it makes a copy of the DNA for the new cell. So what we're looking at here is the original DNA in the original old cell, and it starts to unwind, and both of the strings unwind. And let's just look at this lower string here. So this string is unwound from the original DNA, and it is joined by a series of these left-handed beads that are coming in just the right order to be hooked up with the old string. And so this tan bead has just arrived, and it hooks up with this auburn bead, and the yellow beads here have hooked up with the green beads on the original string. So the net result after this DNA, original DNA, unwinds is that you end up with 2 copies of the DNA. And again, they are 2 copies with strings that contain the same information but in an inverse or opposite sort of way. One of the copies of the DNA stays with the old cell as it dies, and the new other copy goes with the new cell as it will then be needed to make the rest of the structure of the cell. So that's the copy process of DNA, and it's very important to allow our cells to efficiently copy. Now, the DNA that's 3 billion units long is not just in one single long string. It's snipped into 46 chromosomes, 46 parts, and those parts come in pairs. So we talk about there being 23 pairs of chromosomes or parts of this DNA, with these pairs being similar but not exactly the same. We'll see that in a minute. So these chromosomes exist, and what they have on the chromosomes are these dark areas that I've drawn here. This is a gene. This is another gene, and they have separator regions in between these genes to isolate a gene from the next gene. Now, an individual gene in there might contain the information for your hair color, and you've heard things about recessive genes and dominant genes. And so here, for example, very simply, are two genes that are, say they both happen to be the hair color gene, or one of the genes that controls hair color. And one of the aspects on one of these genes would be dominant, and it would say, no, you're going to have red hair, or no, you're going to have brown hair. And the other gene would be the recessive gene and not allow that color to come into this new being. In any event, that's the general idea, that the DNA is broken into these chromosomes, and these chromosomes have genes and separator regions on them. And something we'll see in the next part 4 will be the importance of the tail ends of these chromosomes, which are called telomeres. But in any event, we have 23 pairs of these in each one of our bodies, and they define your overall characteristic. Now, this is a picture of the human DNA existing in 23 pairs of chromosomes. I'm not a geneticist, but and I hope they forgive me for phrasing it this way, but every time I've seen a picture like this, I've thought of it as somebody's dead worm collection. They caught two kinds of worms about the same species, and they just pinned them on this sheet here. You can see that they are the similar species. But this is the way geneticists portray. It's the actual pictures of chromosomes are more complicated than this, obviously, but this is the generic way that they display them. Let's look at chromosome pair number 1 first of all. You can see that they're similar looking, they're similar shape. They seem to contain about similar gene areas. The dark areas here are meant to mean where the gene is. And they're similar but not identical. They contain similar information, say for similar parts of the body, but they're not identical. They're even different colors. The same holds true for the other pairs of genes here up to at least number 22, where you see similar sorts of structures but slightly different characteristics in those genes. Now, where it differs between male and female is in the 23rd pair. For the female has 2 X chromosomes, and these X chromosomes are along that same sort of a pattern. They're similar but not identical. See, they're similar shape, similar color in this case, and similar sorts of information on them. However, the male 23rd pair are significantly different. The male has 1 X chromosome and 1 Y chromosome, and that Y chromosome and X chromosome are not at all similar. And that's very important for a couple of things. Now, what happens at conception is the mother's ovum selects one of each one of her 23 pairs of chromosomes. Not just any 23, but one of pair 1, one of pair 2, pair 3, and including one of pair 23. And so you can see there are lots of different choices that an ovum can make for exactly which chromosome it takes from an individual pair. It might take the first one here, and the second one, and the next ovum might take the second one, the first one, and the first one. And so each of the eggs would have a different set of these chromosomes, and therefore slightly different characteristics of the child to be produced. And similarly for the male sperm selects one of each one of the male's chromosomes, but when it comes to the 23rd chromosome, the male selects either the X chromosome or the Y chromosome. And when the Y chromosome is selected, the child to be conceived turns out to be a male. That's what determines maleness, this Y chromosome. If the male sperm selects an X chromosome, then it goes with the female's X chromosome and we have a daughter born. So that is a selection process, but what happens most interestingly is that when this conception process occurs, as these 23 sets of chromosomes come from each of the prospective parents, the rest of these chromosomes up through 23 mix and mingle. They use the technical term mix and mingle. These chromosomes pairs pair up with each other and they kind of mix and mingle with each other, and some parts of them are lost a little bit or moved around a little bit. So the characteristics of the child to be born are yet more changed slightly as a result of this mixing and mingling process. You've often seen when you look at children and grandchildren or whatever, oh, he has this person's eyes or has that person's nose or looks like that person. And that's as a result of the chromosomes coming all the way through on a certain family line and retaining some of the characteristics. But yet some of those characteristics are not exactly identical, only with identical twins where they get exactly the same chromosome set on a way when a ovum breaks into two and has these exactly same sets of chromosomes, do you get identical children born. But in any event, the net result of that mixing and mingling is that you get a slightly different individual beyond the contributions of the father and mother. But one place where this mingling does not take a case at all is in terms of the Y chromosome. The Y chromosome does not mix and mingle with its opposite X chromosome. The Y chromosome remains absolutely unchanged for a very long period of time. And of course it turns also that the father is the one who contributes this Y chromosome, and he contributes it only to his male heirs, the male child that he's going to conceive. So the Y chromosome comes father to son to grandson all the way down the line as long as it continues. Whereas this other part of the DNA that we talked about earlier, the mitochondrial DNA that's outside of the nucleus but inside of the cell wall, that is something that is passed from mother to all children. So females pass it to all of their children, but it also has that characteristic of not changing hardly at all when it's transmitted from female to children. So we've got two parts of the bulk DNA of human beings, the Y chromosome in males and the mitochondrial DNA in all people. In particular, we're interested in females that are very slightly changing, and that is going to give us a possibility of having what we call a genetic clock to look for the most recent common ancestor of human beings. So, kind of in summary there, the Y male chromosome is passed male to male, the mitochondrial is passed from mother to all children, and both of these two types of DNA, and only these parts of the DNA, rarely mutate. They don't mingle and mix, and in fact, we know the rough rates at which these two parts of the DNA change from studies of some closed societies where we can measure that they change about this many, and it's of the order of hundreds of generations. Somewhere between every 100 and 500 generations does the DNA of these two types change ever so slightly through a mutation that happens just more or less by chance at some interval in time. That then mutated DNA is passed on to all male or all female or all mitochondrial DNA recipients in the same form until it again, several hundred generations later, mutates. So we have the makings here, and I'll show you here shortly of how that's going to work out. We have the makings here of a genetic clock to point back when the original version of human DNA started. So therefore, we can do a genetic test, and what some geneticists have done is they get samples of DNA from a group of either males or females, and they check to see how much different the DNA parts are. If it were males, they take Y chromosomes and they see how much they differ in this representative sample of living human beings today. And the same thing for females. You get samples from 100, 200, 300 males, and if you disperse these samples or take these samples from around the world, not all just from one location, but some from the United States, some from Europe, some from Africa, some from the Near East, the Far East, the Middle East, and you take these samples all together, you should get a reasonably representative sample of how this DNA has changed over time, because all of these people have different family members, different family trees for which they have experienced these changes in these two types of DNA. So they get these samples. They then measure the amount of variation, and what we know right now is that the variation should be of the order of 2 to 4% per million years. This is a reasonably wide range, by a factor of 2 anyway, but we know that it's roughly very unchangeable, but changes at about that rate in terms of small differences in the DNA. Now the first of these tests, and I'll show you in this version in the plot at the left here, we're looking at time here, where this is today, and we're looking backward in time. Here's an eyeball looking backward in time from a data set, and uses that 2% per million years and 4% million years to get an estimate when the first, in this case, the first human female lived. And they came out with a range somewhere between, what, 140,000 and maybe 270,000 years. This was the first data set that was obtained by Rebecca Kahn at Berkeley, University of California, I believe, Berkeley. And this data set consisted of 147 female mitochondrial DNA samples. There were 145 samples from placentas, afterbirths, and these placentas were analyzed for the amount of change, and there were 2 female stem cell lines that were used in this same way. The samples of these 147 samples, 20 or so were, say, American females, 20 or so were African females, 20 or so were European females, some from the Far East, some from the Middle East, and some from other Papua New Guinea as one of the sources where they took samples. So this is a reasonably well-distributed sample over the Earth and should give an answer. And she got these answers 140,000 to 270,000, some number like that. That was the first test that was done, and I've found about 9 tests done up to about the end of 2011, beginning of 2012. Perhaps there are more, but I found these 9 that we'll see shortly. So there were 9 tests that were conducted, and when you average 5 of the best tests, you find that the first female lived about 175,000 years ago, and the first male lived about 65,000 years ago. Now, a couple of things about these results were disturbing to the geneticists who first performed these tests, even with the first test, where they found somewhere around 170 or 270,000 years ago when the first female existed. They were expecting a substantially larger number. The idea at the time was that humans descended or ascended from chimpanzees, and the answers that we were expecting to get were of the order of millions of years, perhaps as much as 5 or 6 million years ago, when humans diverged from chimpanzees. But instead they got numbers around a couple of 100,000, which were a little bit disturbing. The first few tests that were done with females, and in fact the second test that was done used 3 female mitochondrial DNA samples and 4 female chimpanzees to try to get a clue as to what was going on in the way of this transition to chimpanzees. It actually didn't change the answer very much. It moved it up 30 or 40,000 years, but it still was of the order of 200 to 250,000 years ago that the first female existed. That test was not as unrepresentative as I said there, because those 3 female samples of humans were said to represent 215 females in some statistical way. So, but the net result was that even though they included chimpanzees, they didn't change the answer anywhere close to what they were hoping of to get it up a million years. They just got it up a little bit. I chose to use just the 5 tests for both males and females that did not have any chimpanzees, and that's where these numbers came from, 175,000 and 65,000. Now, if all of the data, all 9 tests were included, it only went up to 210 and 110,000. So it didn't change it very much, but I think these numbers here are more representative. The second thing that disturbed the geneticists when they did the first few tests were done with females, because mitochondrial DNA, as a smaller quantity of DNA, can be more easily tested in the laboratory. So after a while they decided to do some male tests. I said, well, let's just check and make sure we get the same answer for this first male human. as we found for this female human. And much to their chagrin, they ended up with these numbers that were a half to a third of the female number. Substantially earlier male or later male finding, and a substantially uh, earlier first female was found. So that was disturbing, and I could not believe that one of the technical papers actually, with this finding, made the joking statement that, well, they couldn't have found Adam and Eve because they never would have had a chance to date. I was just stunned that ended up in a technical paper, but it did. In any event, this difference between males and females has great explanation in terms of historical genesis. Let's look at that. Here is, if you remember from the early on, I think it was in the first part, where we looked at a tree of life, where we had the first single-celled animal here. The animalcule was down here, and then we had all the various animals that have evolved one from another according to evolution theory, with aquatic animals up toward the bottom and mammals in the middle and humans at the top. Well, here's a play on that same sort of thing. Let's look at the male tree of life, with Adam as the first male who appeared on earth, and then as he had children, Seth and Cain and Abel and other sons. With time they had sons, and this tree of male life continued to spread with slightly different Y chromosomes as one of the individuals experienced a mutation. He then passed that mutation on to all future male generations in his line. However, the Bible says that at the time of Noah, that humanity had reached such a sorry state with sin and idol worship and all kinds of breaking of the laws of God, that God sent a flood, and everyone except except Noah and Noah's 3 sons and their 4 wives were the only humans that were on the ark. So all the rest of the humans had perished in the flood, and what that means from us for the Y chromosome, all of the rest of the Y chromosomes in case of males perished at the point of the flood. The only Y chromosome that was present was that of Noah and his 3 sons, which I just said every couple of hundred generations does the Y chromosome change. So very likely Noah and his 3 sons had absolutely identical Y chromosomes. Then with time, as Noah had sons and grandsons and all the rest of the humans that are now on earth, we in effect got a distribution of males that find amongst us males living on earth today that we don't have a lot of variation in a Y chromosome, or at least we don't have as much variation as would be expected if we saw back to Adam. So in effect, the males on earth today point back to the first male that our genes can see, or our Y chromosome can see, is that of Noah about 65,000 years ago. The signal from Adam has been lost. In effect, I don't mean this irreligiously or anything, but the Noah is in effect a new Adam, a new first human, at least from the prospect or aspect of the first genetic person that the males living on earth today can point to. So that's an explanation why you get a substantially earlier value for the male first human ancestor. However, for the females, we have a similar tree here for females with Eve at the base, and at the point in time of the flood, all of the rest of the female signal was not wiped out. The 4 females that were left, the 4 wives of the 4 males on the ark, were not said to be cousins or sisters or close relatives, and so they quite likely represented a wider breadth of the mitochondrial DNA that started out with Eve. And so they point back to an earlier time. I've shown it here. I've just kind of whited out some of the areas of the tree and show that when some of those females died, that some of the mitochondrial DNA signal was lost. It's not an accurate picture of it, perhaps, but I think you get the general idea that female mitochondrial DNA points back to a more ancient, a more distant first female, whereas male DNA now is incapable of doing that. We can only see back to this first male in recent times that we call Noah. So that gives an explanation from the Bible as to why genetic tests for males and females are different. because of the loss of male Y chromosome information at the time of this flood. It gives us evidence that quite likely something like the flood actually did happen. Now, the 175,000 years ago from the time when Eve and quite likely the first male Adam existed is substantially different from what you would get if you read the Bible in the first part of Genesis and you looked at the genealogy that was contained therein. It talks about Adam being the first male born on earth, and then Adam had various sons. One of them was Seth, and then Seth had a son Enosh, and Noah was the son of Lamech, and Jesus was the son of Joseph. And there's a whole series of sons and fathers described in the Bible. If you interpret that word where it talks about son, at least in the English translations, if you believe that's an actual son, and in some cases it is a son because it gives the year in which that son was born, quite likely therefore is an actual son. If you look at that genealogy from Adam up until times that we know about, such as Roman times, around the time of the birth of Christ, you can end up with an estimate for when Adam first appeared on earth. And those numbers come out to about 5773. Less than 6,000 years ago, Adam was the first male on earth if you use this calculation. Now, the Jewish faith uses this calculation, and they interpret this year, or believe that this year is the year 5773, the years since Adam. And that is an estimate. It's quite a bit lower than the 175,000 years that we just saw, but there is some difference here that we can point out. First of all, the word ben in ancient Hebrew is the single word that is used for son, grandson, great-grandson, great-great-grandson, or beyond. So the word ben really just means a male heir. It says, for example, that Noah was a male heir of Lamech. He may not necessarily have been the father of Noah, but could have been a great-great-great-great-great-great-great-grandfather or some such of Noah, but he was a male heir of Noah. Now, other Hebrew texts that people have found outside of the Bible, turns out they list every fifth or sixth generation in the other Hebrew texts. So it's possible that in this case, the Bible, though it's literally true that these are male relatives, would only mention the very important relatives that appear in this scale. And so the number 5773, say 6,000, could be 5 or 6 times greater. It could be 35 or 30 or 35,000, 40,000, some number like that, if these indeed are not exactly fathers. Now, if you understand that, fine. If not, let me just give you a brief analogy. I'm a Green Bay Packer fan, and if I were to name the quarterbacks for the Green Bay Packers, I would list Lambeau, Curly Lambeau, for whom the field where Packers play is named, Bart Starr, Brett Favre, and Aaron Rodgers. I would give you that list of 4 quarterbacks because in my lifetime, my experience, those are the most important, experienced, contributing quarterbacks that the Green Bay Packers have known. I would have skipped about 60 quarterbacks. There are about another 60 quarterbacks who have started at least one game for the Packers, but they didn't last long, didn't do great service to the team, and so I just named the important ones. Well, quite likely that same thing could have happened in the writing of Genesis. It's not an untrue text, but it doesn't mean exactly son, it means male relative. And so I say that at least biblical genealogy is going to higher numbers, and indeed that 175,000 year number could quite likely change, maybe come down a bit to be more in agreement with something like a biblical estimate of 40 or 50,000 years ago that Adam and Eve first walked on earth. I just point this biblical side just to show you that there's nothing irreligious or sacrilegious about finding 175,000 years ago that Adam and Eve existed, in my opinion. It's just a different estimate and may or may not be the right answer, but is at least as an answer. So let's look a bit more at the flood of Noah. We've just seen genetic evidence pointing to the fact, or at least it's consistent with Noah having been about 65,000 years ago. In fact, there's There's evidence that we found a first male anyway, the first male ancestor about that time, and that's consistent with the flood story of Noah as we've just described. His story is contained in the chapter 7, verses 1 through 24, telling how Noah built that boat and how he outlasted with his immediate family and animals from the area on his large ship. There are questions that remain about the flood to this day, of course, and there's work that's been done over the years to try to answer some of these questions. What about the very existence of this flood? Was there a flood at that time? Have we found any scientific evidence? We'll look at a little bit of that. Did it actually happen over the entire earth, over part of the earth? Is another question. The Bible talks about the ark coming to light on Mount Ararat, and if it did indeed come on top of Mount Ararat, that's quite a tall mountain, as we'll see in a minute, and it would have taken a lot of water, and that ark, the remnants of that ark, should be somewhere up on top. Was it localized, or was it really just a localized flood in the Mideast? It was just around the area where all humans, as the Bible says, were at that area at the time. Is that the only place where the flood happened, a localized flood? If so, less water would be required. We'll talk some more about that. Remains of the ark I've already mentioned. Is it on the top of Mount Ararat? Is it some other place? No one has found it. Have geologists found a silt layer making from this worldwide flood? There should be at least a small silt layer throughout the entire geology of the earth. And have they found it? No, they haven't. And then there's yet another controversy about how could such a large wooden boat withstand the ravages of water. Some engineers that deal with ship design feel that it would have broken apart due to the ravages of water. You couldn't build a wooden boat that large. Some of these 6 questions have a little bit of information I can answer, and let's just look at those. Some light has been shed on question 2 about the final resting place of the ark being on the top of Mount Ararat. We know that at least hydrologists and geologists estimate that there's not enough water in the world on the earth today to have reached up to the top of Mount Ararat, which would be about 16,854 feet. And so there's controversy, there's not enough water, so therefore that might lead you to believe that the flood didn't happen. Well, what has happened in recent times is that it has been pointed out that the ancient Hebrew reference to Mount Ararat actually is a reference to the Ararat mountain range. It is not Mount Ararat, but the mountain range of Ararat. It's kind of like the Rocky Mountains in the U.S. or the Himalayas in other parts of the world, where the ark is said, therefore, to have landed on the Ararat mountain range. So it could have landed on a lesser peak. There could have been enough water to cover that close to that peak and other peaks around it, and it could have been a flood that was over less than the entire surface area of the earth. So that's interesting information. There also, in terms of being a localized flood, there has been some geological evidence that has surfaced. It's only been about 7,500 years ago, but there has, 7,500 years ago, it has been found from geologic results that there was a very rapid flooding of the Black Sea from the Mediterranean Sea. The Black Sea during a certain climactic period was basically dry, and after an ice age with all kinds of melted water, that melted water all of a sudden broke through, flooded the Mediterranean, and the Mediterranean overflowed into the Black Sea. So that at least there is physical, here is a physical example of how a rapid influx of water could have occurred. The Bible says that it was rain, but here is the case where it could have been, or a case where it was filled by just a large influx of melted glacier water. So there's a little bit of evidence there. Now, I remember in my youth of seeing National Geographic films and studies of the idea that they were looking for the ark on the top of Mount Ararat, and they were 15 or 20 miles away looking through a telescope, and they saw an outline of the cross section of the ark, indicating that maybe that was the ark had broke up, but the weather was severe they couldn't get to it, and by the time they got to it, avalanches and things had covered it. So it has never been found at this point. We may continue to look for it, I'm sure, but whether it's found on top of Mount Ararat or someplace else or not found is a question that has not yet been answered. And finally, in this idea that you couldn't build a boat large enough, as it's talked about in the Bible, I'm not a nautical engineer or someone who designs or builds boats, but the one thing I do know is that wooden boats would float on rising water, and the flood, as described in the Bible, is not talked about as a violent pouring in of water or movement of water. Just talked about a lot of rain where the water accumulates over a relatively quick period of time. And I know that wooden boats float on rising water, so I don't have any problem with the idea that a boat could have been built by Noah and existed during the duration of the flood until the time when the flood waters went down. So let's look briefly at the conclusions we've seen here in part 3 about Adam and Eve and Noah. That Adam and Eve lived about 175,000 years ago according to the genetic test results on mitochondrial DNA, so they were the first humans to appear on Earth, and that same sort of analysis led us to conclude that quite likely the first male that was found was Noah, because all other Y chromosomes were wiped out at the flood. And it enables at least the initially confusing results that the geneticists found for these two different times for the first female and the first male. And then we've just seen, at least with the flood of Noah, that it can be interpreted as being a localized flood just in the Middle Eastern region, with the ark settling on one of the lesser mountains of the Mount Ararat range. There is no actual concrete proof on this part of discovering exactly where Noah's boat ended up and where the flood happened, whether it was local or worldwide. But I think my own particular opinion at this time is that it is a localized flood just in the Middle Eastern region. Next time in part 4, you will see some explanations of the 1,000-year lifetimes in the human races that are other parts of Genesis that might be a little bit unbelievable to you, even if you accepted the first parts 1, 2, and 3 results. I'll conclude with part 4 here to show you how 1,000-year lifetimes are certainly scientifically possible and how vitamin D can explain the origin of the human races.